Okay. Thank you, everybody. I'm just going to talk. My name is Catherine Berman. Laura Hertz is presenting with me as well. If you have more questions about the fluoride riboswitch, please find her. But in the spirit of switches, the talk that we just saw, our poster is also about switches. So our lab, the Lux lab that I'm a part of, I'm a graduate student in that lab, we study co-transcriptional folding of RNA in general. And one of a very convenient model for studying co-transcriptional folding are riboswitches, which riboswitches are naturally occurring RNA aptamers that probably many people here know and love. And they confer the ligand binding, they communicate that with an expression platform. And our talk, or in our, the unifying idea of our poster is kind of finding this pattern of this very, very kind of beautiful strand displacement mechanism in very different riboswitches. So a riboswitch, the fluoride riboswitch, which is a transcriptionally regulated riboswitch, and then the TPP riboswitch, which is a translationally regulated riboswitch. So thank you very much. Did I come in under 90 seconds?
Yes, great.
Cool.
So I'm confused. The same posters for Laura or not?
Yeah.
Okay.
Yes, it is.
Okay.
But I'm just talking.
Oh, I see. Okay. All right, great. Thank you so much. And so, yes, as last time, we will hold our questions till the end. And if you have posters in person, we can also look at that. Okay, so next, sorry, the sharing is very, there we go. All right. So hopefully everyone can see the full poster. So we should have, I don't see the speaker in the chat. Nope. Okay. All right. So this is supposed to be Emily Chow. Are you in the audience?
I am here.
Oh, so, you're here.
So hi, everyone. So I'm the first year graduate student at York University. So you won't be able to see my poster, but the goal of my project is to design a bifunctional aptamer that has positive cooperativity, because we believe it, oh, there it is. So it will be, because it will be more sensitive in biosensor application. So I'll be using ITC to quantify the interaction between quinine and aptamers. So that's not mine, but like, okay. So this is the scheme of MN25, a variant of the cocaine binding aptamer. It is being used because it has an extensive at the 5' end, and that is complementary to another MN25 molecule. So MN25 is loosely binding at free state. So it is structured when the ligand induced folding mechanism with a short stem, and it is pre-structured in the long stem one. The blue circles represent quinine as the ligand. So upon quinine binding at one site, another MN25 should associate and form an interhelix aptamer, and this formation should structure the MN25 molecule. So the second KD should be tighter than KD1. So as a preliminary work, we look at this aptamer, OR9 and OR10, which OR10 has longer base pair on stem 1 and got some good ITC result, but we will still working on getting the MN25 result. Thank you.
Great. Okay.
Hi. So this is my poster. Hi, everyone. So my name is Mujab Shukefe. I'm a PhD student from the University of Bonn. So here we identify a clickmer that can recognize BC3 cells. through split combined cell helix. So the idea of the split combined is to split the starting library into 5 parts, functionalize each part with one functional group, and then combine the 5 parts and start the selection. So during the selection, different counter selection steps were included to reduce the unspecific binders. And at the end of the selection, we were interested in one of the clickmer. So this clickmer showed binding with 4 out of 5 functional group. So in this poster, we focus only into 2 functional group. So the idea here is like if we functionalize the aptamer with one functional group, it will recognize only BC3 cells. And if we functionalize the same aptamer with another functional group, it will recognize also MCF7 cells. So we confirmed this by doing an internalization study. And if you want to know more about the selection procedure, clickmer, you can visit my poster. Thanks for listening.
Great job. All right, so next we have, oh, sorry, there we go. Hopefully everyone can see that.
Perfect. Can you hear me?
Yes.
Awesome. So using NMR, we measured the exchange rate constants for the amino protons in the cocaine binding aptamer. This allows us to look at the dynamics of individual bases and base pairs within the aptamer and understand how they change. This was done at various temperatures ranging from 5 degrees to 45 degrees for the free and ligand-bound aptamers, and with 2 different cocaine binding aptamer variants. MN4 is a long stem 1 construct, which is folded in the absence of a ligand, while MN19 is a short stem 1 construct, which is only partially folded in the absence of a ligand. So on the top right, I have a dynamic secondary structure figure. The redder a base pair is shaded, the more dynamic it is, whereas the bluer a base pair is shaded, the more rigid it is. For both aptamer variants, we see a reduction in dynamics at the 3-way junction/ligand binding site upon binding of either ligand. Outside the ligand binding site, we do not see a huge change in the dynamics of each base pair. Quinine, which binds about 50 times tighter to the aptamer than cocaine, but we do not see a huge difference in the dynamics between the 2 ligand-bound aptamers. Between the 2 aptamer constructs, the short stem 1 variant is more dynamic than the long stem 1 variant. This is seen in both the free state and in both ligand-bound states. If you'd like to know more, please check out my poster online. Thank you very much for listening.
Wonderful. Everyone's doing a great job. Oops. All right. Sorry, not switching. There we go. All right, Ismaili.
Always takes me a while to make it up here.
Of course.
Hi, everyone, and good afternoon. My name is Nima, and I'm a first-year PhD student at Limes, University of Bonn. We have recently developed an aptamer binding, bearing indole modifications that binds to SARS-CoV-2 spike protein and its variants of concern. We implemented a split combined approach, as my colleague just explained this approach, to screen for modifications mediating best binding. And the library was functionalized by click chemistry, copper catalyzed azide alkyne cycloaddition. After 8 rounds of selection and round by round deep sequencing of the libraries, we obtained and characterized our top clickmer candidate, which showed interaction with COV2 spike protein at low nanomolar affinity. However, rapid emergence of viral variants has made it crucial for our work to constantly investigate the recognition capability of our click map. So we assayed the interaction against several variants of concern via flow cytometry, SPR, MSD, and verified binding to alpha, delta, omicron, and mu variants. And our preliminary investigation of the neutralizing potential of this aptamer has indicated reduction of infection of Vero E6 cells and against Delta pseudovirus. So the current step for us is focusing on a series of plaque assays using real virus, Delta and Omicron variants. and to gain some thermodynamic knowledge and wrapping up our truncate studies. Thank you for listening. And if you have any questions that are interested, please contact me.
Thank you so much. That was great. 1 second. All right. So who do we have now? I can't see. All right. Eunice Kayam. Please go ahead.
All right. So my name is Eunice Kayum, and I also get to work with Professor Johnson at York University. I would like to share my current research on a dopamine binding aptamer and my investigation in its binding mechanics, as this can provide an insight into how a prevalent G quadruplex structure is able to give rise to affinity and specificity in aptamer function. I primarily work with a variant referred to as the MUD3 variant, which is proven to have binding properties similar to that of the original, in addition to consistently being in one conformation observed with NMR. Prior research has shown that this aptamer forms a 2-tetrad G quadruplex upon binding dopamine, and thus to determine which guanine bases are directly involved in this, we performed a series of ITC experiments on site-specifically mutated variants of the MUD3, replacing guanines with adenines. Under the assumption that the G quadruplex facilitates the binding to dopamine, we hypothesized that 8 mutants would lose functionality. Upon reviewing the data, we identified 8 Gs labeled here in red, although it's kind of small, but it's on the bottom left there. 8 Gs did eliminate all binding. The other 3 Gs did show some reduction in binding. We then looked into simplifying the structure by truncating it from the long stem in order to make the assignments of NMR spectra less ambiguous and simpler. We determined that the del7 variant reflected an acceptable balance between function and simplicity in order for further investigative techniques. We see a general trend as the KD trends up, the more truncations applied to the aptamer. However, when consulting the NMR spectra, we see a consistent spectra change in the non-Watson-Crick region in the bound and unbound form, from which leads us to believe that the del7 retains the ability to form a G quadruplex and subsequently bind dopamine. Further two-dimensional NMR studies are currently underway on the del7 variant in order to determine the exact geometries and spatial configurations of the aptamer and its substructures. If you have any questions regarding my research, please feel free to approach me at my poster. Thank you.
Wonderful. All right. So next we have, apologies, everything on my screen blocks the speaker's name. Lisa Ledortz.
Yes. Good evening, everyone. I am Lisa Ledortz, a second year PhD student in BIPA from France. And my PhD project consists in developing aptamers against the zoonotic and tick-borne bacterium Anaplasma phagocytophilum. The first step was to set up a successful SELEX protocol. And as you know, SELEX is associated with a high failure rate, and the generation of ssDNA is the most crucial step. Several methods currently exist, whether very few papers compare them. So we compared the quality and quantity of ssDNA generated after the two most widely used methods, so capture on streptavidin beads and lambda exonuclease digestion, before and after purification step. In addition, we investigated the performances of free ssDNA quantification techniques, so qubit, nanodrop, and gel-based quantification. And our results demonstrated a higher reliability of the gel-based quantification and confirmed the superiority of lambda exonuclease digestion. Our study will help scientists to implement a difficult but essential step for successful aptamer selection. And we used our findings to select aptamers against Anaplasma phagocytophilum. And so far, we have a promising result since a rapid and specific enrichment of potential sequences was observed by qPCR and NGS. So thank you for your attention, and my poster is available for more details.
Great, thank you so much. All right, next we have Tyra Lewis.
Okay, hi. So my name is Tyra, and I'm a master's student at Trent University. And this work introduces a localized surface plasma resonance aptar sensor that is designed for the detection of the SARS-CoV-2 spike protein. So essentially, with the increased transmission of the coronavirus came a corresponding increase in clinical testing demands that are struggling to be met in these times. And even though conventional testing methods that target antibodies or nucleic acids have been heavily relied upon during the pandemic, these techniques are not ideal for quick response times or early detection. And so this work demonstrates an alternative method to testing, where using LSPR, we are able to achieve rapid viral detection by targeting the virus's spike protein with the use of a single-stranded DNA aptamer, which we refer to as the S1 aptamer. And our aptasensor is fabricated in a 2-channel system using high-affinity streptavidin biotin chemistry. And when compared to other surface proteins of the coronavirus, the S1 aptamer shows selective and high-affinity binding to the S1 protein, as you can see in that bar graph on the right. And the protein levels can also be quantified with a limit of detection of 0.26 nanomoles. And so to summarize, the aptasensor also allows us to monitor sensor fabrication and binding of the molecules on the gold nanoparticle surface. And we can also achieve the desired shortened processing time, great selectivity, and real-time measurement, which is great for point-of-care testing. Thank you.
Wonderful, thank you. Okay, so next we have Cynthia Martinez Liu.
Yes, good afternoon. My name is Cynthia Martinez, and I will present the work I'm currently working on Mexico, and which is based on the design of aptamers that recognize and differentiate between the viral protein RBD, the SARS-CoV-2, and H1N1 of influenza virus to improve the diagnosis of respiratory disease. For this purpose, we designed a library were incubated with decreasing concentration of target protein to perform positive selection, and subsequently each pool was subject to 3 cycles of negative selection using BCA and either protein H1 or RBD. And each pool was cloned in vector, topo vector and transfer. And I'm currently working in the sequencing. But the aptamers obtained in this work will be used to improve the detection of respiratory disease, using them in various detection systems. And in addition, for characterize the design library, the amount of single-strand DNA recovery after each positive selection cycle was quantified by real-time PCR, and the library was found to behave differently when subject to the different target proteins. And finally, we are working in a detection system in this respiratory disease with these aptamers. And is all this question is welcome. Thank you.
Great, thank you so much. All right, so now we have, now we have Megan Osborne.
Hi, everyone. My name is Megan Osborne, and I'm from the Johnson Lab at York University. I'm currently working on the MN series of cocaine binding aptamers with varying STEM1 and STEM2 lengths. And the goal of my project is to determine an effective aptamer to have the most effective biosensor. I'm looking at the stability and volume change of these aptamers, as little is currently known about these parameters and the alterations with ligand binding. This will be done through DSC, differential scanning calorimetry, which scans the heat flow in and out of a sample as a function of temperature. It can also be used to give thermodynamic properties and the pressure perturbation experiments, which will monitor the change of volume with pressure pulses as a function of temperature. MN4 is currently the aptamer for which I have a full data set, and there was a melt temperature change from 52.55 degrees Celsius to 65 with binding to 2.2 millimolar quinine, with no change in enthalpy, while the change in volume at 90 micromolars of bound and free were perfectly superimposed onto one another, though a slight volume change can be seen at 62 degrees, which will need to be investigated further. Thank you.
Wonderful. Okay. So now we have Sophie. Go ahead.
Yes, okay. So hello, everyone. My name is Sophie Skelink, and I'm a PhD student in the NMR STL research group of Professor Martens at Ghent University. Most of you already know of the potential of DNA aptamers as small molecule bioreceptors in sensors, but probably also know that many challenges still remain to be addressed for these to break through in real-world applications. And one of those challenges is to address the lack of insights at the molecular level when talking about factors determining sensitivity, selectivity, and affinity, as well as that we want to try and go beyond that cartoon-like approach when describing the underlying sensing mechanism. So our poster describes our efforts to contribute towards this goal. Specifically, we are developing NMR-based strategies. And for this, we here use the testosterone binding DNA aptamer called TES1. So you will see that we devised a titration protocol to handle the low solubility of our target testosterone. We also did several complexation studies for, on one side, the original sequence and also a more NMR optimal sequence, which was obtained by trimming of the duplex as well as introducing some nucleotide substitutions. And we also investigated the structure switching mechanism of this sensor under NMR conditions. So if you are interested and you want to know more, feel free to take a look at my poster, and I'm available for questions. Thank you.
Great, that's wonderful. So I think, Erin, you were going to share yours next. Erin Shora. On his way up, perfect.
You have to.
Do you have the slides there? Do you want me to share them? Are the slides there, or do you want me to share them?
I don't have it here. I think they are trying.
Oh, okay. Oh, there you go. Great. All right, anytime. Go for it.
Mhm. I see it here, but it's not on the screen. All right. All right. Hello, everyone. Thanks to organizers for the opportunity. Today, I'm presenting my results showing first visible fluorescent probe for tribate junction. Next, please. So first, we wanted to understand how ligands interact with tribate junction. They interact with tribate junction structure. And our collaborator, they synthesized this dye in the lab, and they noticed that this dye interacts with cholic acid aptamer tighter than its ligand. So we wanted to see if this dye would interact with cocaine binding aptamer as well, because they both, as you see here, they have tribate junction structure. And here's the structure of the dye. You see here is the unbound and unbound structure, and here's the bound after pi-pi interaction. Next. Can just scroll next one. All right. So here is the results after. Here's the visible the UV visible, and we see that there is a shift on the emission, and we notice that the emission of the dye versus unbound dye and aptamer is like 600-fold increase in the emission, and also the quantum yield increased 10-fold. And also we notice the fluorescence anisotropy of the dye. When we titrate the aptamer, we are able to detect the KD, which is comparable with ITC method that we tested already. And we use ATP aptamer for the negative control. Another thing we notice is that we can use quinine, which is a tighter binding ligand, and use it for a competitive binding model, use it to displace the dye from the complex, and we were able to get the displacement constant. So for future studies, we are exploring more additional myrcene dyes, and we are investigating their activities in vivo. Thank you very much for your attention. If you have any question.
Great, thank you so much, Aaron. All right, so now we have, now we have Julia. It's not displaying yet. I'm not sure why. There we go. Go ahead, Julia. Okay, so I don't see Julia in the audience. We'll go to the next one. So now we have Paul Combs. Do we have Paul there or Julia there in person? No. Oh, he talked this morning. Okay, so there's just a mix up on the one sent to me. No problem. Okay, and then let's see. We have also Erin Giroux.
Hi, everyone. I'm Erin. I'm an undergraduate student at Trent University, and I've worked on a project with my colleague Tyra, who presented earlier, using the same aptasensor design for the SARS-CoV-2 S1 protein. But this poster specifically focused on our project to functionalize this LSPR sensor with our streptavidin and biotinylated aptamer outside of the instrument, which we refer to as offline rather than the traditional online methods inside the instrument. So this preparation method allows us to use smaller volumes of the reagents and increase the number of sensors that can be fabricated at once. So since these sensors are fabricated outside the instrument, they can be prepared and distributed as ready-to-go kits, sensor kits for consumers. So this would allow a user to dock the prefabricated sensor into the instrument and introduce their sample almost immediately without the need for any preparation steps and achieve results in about 10 minutes. So this design provides a rapid and sensitive and simple alternative to current gold standard laboratory detection methods for SARS-CoV-2, such as PCR. Thank you.
Beautiful, thank you so much. Okay, so I think we just had one poster that didn't make it. So Martinez, Roque, if you want to share your screen, you can go next.
Can you hear me?
Yes, and it looks good.
Thanks. So hello, everyone. Thank you for your attention. My name is Mateo Martinez, and I am a doctoral researcher in the final semester of my PhD at CIMBESTAF, Mexico. My project focuses on the selection of specific aptamers against the spike protein of the SARS-CoV-2 virus. So as we all know, the rapid spread of COVID infection throughout the world led to a global public health and economic crisis, triggering an urgent need for the development of low-cost vaccines, therapies, and fast, high-throughput detection assays. Therefore, together with a multidisciplinary team from different research institutions, we employed capillary electrophoresis and next-generation sequencing to identify aptamers that then were used to develop two types of biosensors, sandwich type fluorescence aptasensor and an electrochemical aptasensor. We believe that aptamers have great potential in the development of better biosensors as diagnostic tools, since they have lower fabrication costs, have higher thermal stability, and are easier to synthesize with higher reproducibility compared to antibody-based tests. If you have questions, please approach to me during my poster presentation. Thanks.
Great, thank you so much. Just really want to make sure there was no one else who was missed. If there is, come quickly to the podium or in the chat. And if not, I'd just like to give everyone a round of applause for those great flash talks, lots of great information. For those posters that are in person, I'm jealous I'm not there, but all the posters are available on the Aptamer website for all the participants. And thank you so much for joining us for this virtual session, especially those of you in person handling all the technical difficulties that we have. So that wraps it up for this day. I'm not sure if Mohammed or anyone wanted to say anything. I bet everyone's eager to go to their dinner and see you all tomorrow.